For decades, we've been in a fight against cancer. But what if I told you we've been fighting with a very blurry view of the battlefield? Well, today, we're going to look at a groundbreaking new approach that's creating a far more detailed map, one designed to find all of cancer's hidden weak spots. So, to really get why this new map is such a game changer, we first have to wrap our heads around the huge problem that researchers have been facing. As it turns out, our entire arsenal of targeted cancer drugs is aimed at a surprisingly, almost shockingly, small number of targets. How small, you ask? Well, get this. If you look at all the different types of cancer and all the targeted drugs the FDA has ever approved, they all go after fewer than 200 unique proteins. That is it. Fewer than 200 targets to fight one of the most complex diseases we've ever faced. And that, right there, raises a massive question. Why are we using such a limited toolkit? This has been the central bottleneck in developing new, more effective cancer therapies. Finding good new targets is just incredibly difficult, but that might all be about to change. So let's meet the hero of our story, this powerful approach called proteogenomics. It's a technique that offers a solution by giving scientists a much, much deeper view inside a cancer cell than they've ever had before. Now, the name sounds a little complicated, I know, but the idea is actually brilliant in its simplicity. Proteogenomics basically combines two incredibly powerful fields: genomics, which is the study of our genetic blueprint, and proteomics, the study of the proteins that actually do all the work in our cells. By smashing them together, we get a radically more complete picture of what's going on. Think of it this way, okay? Genomics, our DNA, it's like the blueprint for a car. It shows you all the potential parts, everything the car could be. But proteomics, that's like popping the hood while the engine is running. It shows you what the cell is actually doing right now in real time. And that, as you can imagine, is absolutely crucial for finding drug targets that will actually work. And the scale of this research is just immense. We're talking about scientists integrating data from DNA, RNA, and proteins from over 1,000 patients across 10 different types of cancer. What they built is pretty much the most detailed, multi-layered map of cancer ever put together. So, you've got this incredible new map. Now what? How do scientists actually use it? Well, let's dive into how they can now hunt for cancer's vulnerabilities with a kind of precision they've never had before. The researchers used three main strategies. First, they looked for the cancer cell's addictions. Proteins that are way overproduced or just stuck in the on position. Second, they exploited a fascinating weakness called synthetic lethality, and we'll talk more about that in a second. And third, they looked for unique flags on the cancer surface that our own immune system could be trained to attack. Okay, that second strategy, synthetic lethality, is a really clever concept. Let's use an analogy. Imagine a person with a broken leg. Now, they can still get around, but they become completely 100% dependent on a crutch. This is exactly how the strategy works. A cancer cell often breaks its leg by losing a key protective gene, like the famous tumor suppressor TP53. That loss forces the cell to become totally reliant on a different protein. That's its crutch, just to survive. Now, we can't easily fix that broken gene, but what we can do is design a drug to kick away the crutch. And when you do that, the cancer cell just collapses. And this isn't just theory. The study found a perfect real-world example. In breast cancer tumors that have lost that protective TP53 gene, the activity of a partner protein called CHK1 just goes through the roof. The cancer cell is now addicted to CHK1, and that makes CHK1 a perfect, high-priority target for a new drug, but only in patients whose tumors have that specific weakness. Okay, so the theory is elegant, it's smart, but does this whole proteogenomics approach actually work better in the real world? The researchers knew they had to put it to the test. This is really the million-dollar question, isn't it? It's one thing to come up with a new method, but it's a whole other thing to prove it's genuinely better at predicting which drug targets will actually make a difference. And wow, the results here really speak for themselves. This chart shows you the accuracy of predicting which targets would actually respond to drugs in lab tests. The older methods, which only look at gene dependency, they were about 57% accurate. Not bad. But the new proteogenomics method, the one that layers in all that protein data, it jumped to 76% accuracy. I mean, that is a massive leap in predictive power. Let me put that another way, because this is the key point. Using this integrated proteogenomic approach made it 2.6 times more likely to correctly pick a drug that would successfully kill cancer cells in lab experiments. That means so much less time and money wasted on dead ends, and a much faster path to therapies that could actually help people. So, what does all this mean for the future of treating cancer? Well, it means we're moving from a blurry sketch of the battlefield to a high-resolution, multi-layered, interactive atlas. And hey, don't just take my word for it. The authors themselves, writing in the journal Cell, said this work provides a comprehensive landscape of targets. It's a resource built from the ground up to speed up everything from finding new uses for old drugs to developing brand new therapies. So, the big takeaway here is that this single study provides a true treasure trove. We're talking a rich list of new targets for future drugs, a playbook for repurposing existing drugs for new cancers, a catalog of biomarkers to better match the right patient to the right treatment, and maybe most importantly, it's a public resource that researchers all over the world can now use to supercharge their own discoveries. And that's the final, most powerful point. This isn't just about one discovery or one potential new drug. The researchers here have created a foundational new atlas. It's a tool that's going to guide scientists for years and years, potentially leading to hundreds of new ways to fight cancer. And it all comes from finally being able to look beyond the blueprint and see the actual running machinery of the disease.